Hello everyone, my name is Alex Ceccaldi. I'm the General Secretary of the ETPN, the European Technology Platform on Nanomedicine. Today is the third session of the Metrino Academy. It's really a concept that we came with to try to take the best out of this European project, Metrino, and give it to you, to give back to the community. So it doesn't say, it's not just a set of results, research paper, PDF guidelines, but something really tangible for you. So it's a series of webinars, today the third one, and I propose that maybe we start sharing our presentation, please. So I guess you all know the Metrino Academy right now, if you are here, but maybe not. So it's a monthly webinar series designed to bring very practical, usable guidance to the European nanomedicine ecosystem. So today, I guess you all know we have two great speakers. My dear colleague Clémence, we have the opportunity to present them and introduce them correctly in a few seconds. But just in a nutshell, today we are talking about a very hot topic in nanomedicine indeed, RNA-LNP, but not the usual way to talk about that, not just buzzwords or hype, to do it the proper way, how to correctly synthesize, measure, but also compare your measurement with other labs to be sure that you're doing it the right way, which is not that easy, as you will see, but we have fantastic experts to help us to do that at least better, if not perfectly well. So the agenda is quite simple. We start by a very short introduction to explain a little bit what is Metrino and how we'll work this session today. Please pay attention to what my dear colleague Clémence will tell to you. The main part is indeed the talks that we will hear from Enrica and Jérémie about standardizing RNA-LNP measurements across different labs. And finally, we call it the best part because it's supposed to be the best part. It's up to you to make it a good part. It's the Q&A. We really wanted to make it very interactive. And as Clémence will explain, you can ask your question whenever you want, but we will take the time to properly discuss them at the end of this webinar. But now, before we move on, I'll hand over to my colleague Clémence.
Hi, everyone. So I'm Clémence Nadal. I'm the new European project manager at ETPN. And I will start just right now with explaining a bit of housekeeping rules, just so I know it might seem a bit boring or starting with rules, but it's just in order that the session goes as great as possible. And so of course, you are welcome to have your cameras on, so it's going to be super friendly. You will have your microphone off by default, and the chat will be disabled just because we want you to focus on the presentation. But at any time, if you want to ask a question, there is the interactive Q&A tool. You can ask the question whenever you want, and you can also vote for people's question. And during the Q&A, we'll start asking the question with the most votes. So please note that this session will be recorded, and later we will share the replay with you. But first, I would like to explain a bit why did we create an academy for Metrino. So we know that it is a complex topic. Nanomedicine is complex and metrology as well. So we feel like there is a need for pedagogy, and an academy is just perfect for that. So the concept is simple. Each session we are focusing on one topic. So one topic in nanomedicine, one nanomedicine challenge. And we want you to leave this session every time with something very actionable that you can use on the next day in your work. Because we know that this kind of pedagogy needs some regularity, we decided to make it a monthly community rendezvous. So I guess lots of you know what Metrino is, but for those who don't, I'm just going to explain a bit who we are and what we do. So Metrino is short for Metrology for Innovative Nanotherapeutics. And the aim of this project is really to democratize metrology standards in nanomedicine. It's a project that is funded by the European Association of National Metrology Institutes, and it is a large consortium that is led by metrology institutes, SMEs, academical lab universities, and the ETPN Association. The people who actually are leading the project are the work package leaders. So we have Ute in Germany at BAM, Robin and Christian. Robin is the project coordinator. They are both at PTB in Germany. Leonardo from INRIN in Italy, Alexandre from ETPN, and of course, Enrica from LNE, who is one of the speakers today. So now, what do we do? The Metrino has 3 pillars. The first one is we want to provide methods for you. So we developed standard operating procedures for the characterization of nanomedicine. That's the first pillar. The second is the development of a roadmap towards certified reference materials. And the last one, it's exactly what we are building today, what we are doing today. We are trying to make connections between people who usually don't necessarily interact, nanomedicine people, metrology people, and that's what we are doing today, creating a collaborative ecosystem. So a bit more information on the Metrino website. So it's super easy, metrino.eu. Go for it. There is a lot of content. If you want to make yourself a culture about metrology for nanomedicine, it's really the perfect website to visit. So go for it. As Alex said, this is today's session 3. So there was already session 1 and session 2. You can find the replays on the websites. Both were super interesting. With high quality content. The first one was on the industrialization of nanomedicine with nanobiotics. And the second one was about choosing the right measurement method. So if you haven't seen them yet, they are on the website. Finally, just a few words about who you are today. So we are really happy to have over 70 participants. Lots of you, almost 80% of you are from academic institutions. But we also have people from industry, metrology institutes. And we are also very happy to see a big diversity in terms of countries, with over 15 countries that are represented today. And now let me introduce you to the speakers. So first we have Dr. Enrica Alasonati. Hi, Enrica.
Good afternoon,
Hello to everyone.
Hi,
Good
Enrica.
afternoon.
So just a few words on Enrica. She's a senior scientist at LNE, the French National Metrology Institute. She has over 20 years of experience measuring and characterizing nanoparticles in complex samples, and also including micro and nanoplastics. Her work supports environmental studies, consumer product testing, and also regulatory needs. She is involved in a lot of Euromed projects, and today she's here because of her involvement in the Metrino one, of course, and she's also very involved in international standardization. Our second speaker, Dr. Jérémie Parot. Hi, Jérémie.
Hi, everyone.
How are you today?
I'm doing great.
So a few words on Jérémie. He is a senior research scientist at SINTEF in Norway, and an analytical chemist specialized in nanoparticles. He is leading project on RNA lipid nanoparticles from the design and optimization to the production. He is an expert in field flow fractionation, and he develops measurements methods for very precise characterization. And he's also super involved in standardization, including with the ASTM norm committee E56. So thank you both for being here. It's a pleasure to have you today.
Thank you, Clémence.
Okay, thanks a lot, Clémence. Very useful. I can tell you that if you respect the rules and if you get involved, it will be a great success for you all, guys. To me, this is an important session because it's a bit the DNA of Metrino. We have a fantastic nanomedicine expert and a fantastic characterization and metrology expert working together, working on the same presentation, by the way, to provide some knowledge. So I guess now it's high time to listen to you guys. And if you wish, the floor is yours for around 40 minutes. And but we will have some. I mean, you won't be passive, guys. The attendees, you will have to answer some polls in a few seconds from now. Jérémie and or Enrica, the floor is yours.
Yeah, hi everyone. Happy to start. And before getting engaged into the topic, I think, as Alex said, one of the key points and one of the success will be to have a very good Q&A, which I think can be questions, but it can also be feedbacks from users in regarding the methods and techniques we're going to talk about, or just point of views. I think the more the better. So please spam as much as you can the Q&A. So Alex is going to be fighting with the questions to have a good session after that. So to start with, Alex, Clémence already presented us, so it's kind of good. And the topic is tiny particles and big challenges, standardizing RNA-LNP measurements across laboratories. So it's a big title, but a tiny topic. To take back the title, we're going to be focusing a lot on field for fractionation technique and some points on RNA. But please first look into the poll that Alex pulled up.
Yeah, I triggered it a bit fast. I'm sorry, Jérémie, but maybe I can read it so that you get involved. First, we want to know you better, guys. What best describes your role with RNA-LNP? Do you develop and formulate RNA-LNPs? Do you characterize and test them? You can have multiple answers. Indeed, it's not separated and mutually exclusive. I work on payloads analytics mainly. I work on manufacturing, scale-up, let's say industrialization. I build or I supply tools around, let's say, instruments, metrology, etc., or I just follow the field. I'm not really hands-on. Next, what do you measure on a daily basis, or at least routinely, on RNA-LNPs? Guess what? The size and distribution, but also the concentration of particles, the number of particles, their morphology, the payload concentration, the integrity of RNA, maybe some surface properties like the charge indeed, or some stability or aggregation. It's just, let's say, a greet and meet. I leave you around, let's say, 30 seconds, and then I'll show you the results. Interesting results, by the way. And I will show you the results right now. So we have a lot of people in characterization. That's, I mean, good news and bad news at the same time, because we want to have also people that are not experts, but wait and see. Indeed, you also develop, but not that many people working on scale-up. And indeed, what we will discuss today is absolutely essential if you want to scale up at some point in a decent conditions. But we also have people just interested. Guess what? The size is still of interest for some people in LNPs. Who would have told that? But also concentration, morphology. And Jérémie, look at RNA integrity. It seems that we don't care about the integrity of RNA, right? Let's talk about that in a couple of minutes. Now I hand over back to you, Jérémie.
So to start with, let's go a bit into what's RNA-LNP. So mRNA, so we're going to be focusing a lot on mRNA, even if it can cover broader than that when we talk about LNPs. But let's talk a little bit about mRNA and the field. So mRNA is a single-stranded ribonucleic acid transcribed from DNA. It is kind of a magical thing that you can trigger to produce any type of protein within the human body by using the right mRNA sequence, which means that you can have a very broad range of applications looking at it. But the drawback is like it must be used with a delivery vehicle. That's where the lipid nanoparticles are coming in the game, but it's not always the only one used. Some projects can be around like playing with liposomes or lipid polymer hybrid nanoparticle. But the question can be like, if you need a vehicle, the question is like, why? So first of all, you need to protect your mRNA because as if you would inject it IV or intramuscular in the body, it will be degraded. And with mRNA, it's like one break, no effect. So you need to keep the molecule intact. The vehicle will also trigger the endosomal escape. An efficient clearance of the vehicle is quite important. And then to have as much as possible some cell-specific uptake or organ trophism and to have low toxicity. I think we are all aware that the COVID-19 vaccine showed really well to the world that LNPs are essential for stability and intracellular delivery and potency. But that also goes with a big investment and diverse pipeline into many different therapeutic ranges. If we look at that from that perspective, the market is getting very big, and we are expecting above the $1 billion by 2030, generated by the drug market itself, not the investments, with currently 4 mRNA vaccines in the market, 5 approved gene therapy. So the market is really growing. There is a big interest, a lot of investments. But then it goes with like, yeah, okay, I'm developing a product. I want to get the proper methods and the proper critical quality attributes. And when you look at it from a developer point of view, either from an analytical lab or an industry lab, you realize that, okay, you are fighting with like so many different quality attributes. On this graph here, you have like on this table, 23 different critical quality attributes just for the drug product itself, which is about 24 different techniques. So it is a very complicated game in the sense that, okay, you have a lot to look at and a lot of different methods, which come from the complexity of the material itself. So when you look at it, you're like, okay, I can end up with so many different techniques. And the question is like, which one should I use? Which one are easily available? Which method are easily available or easily accessible? Meaning like I can find the detail of the method somewhere. So when you look at that and you think about what you want to do, you will end up with the first question, which are, okay, which technique should I use? Which method? Is there SOP? Or which CQA will that enter? And which data quality I'm going to get? And that's where this webinar is coming into, like the standardization part of it. Because when you think about that, standardization could offer like to have a protocol that will make things much easier for people to analytically talking, analyze your product. And I like to say that standardization isn't optional. It's like the fundament to get reliable quality assessment and to get like easier access to regulatory acceptance. But given that the product is very complicated or very complex, it introduces some unique CQAs not seen in traditional small molecule or protein from a normal pipeline. So it gets a bit more complicated. Regulatory exceptions for validating and reproducible data are high, particularly for emerging modalities. So it makes things quite complex. So shared and robust method reduce like any questions around and makes the field getting faster and better. And also it makes things safer from the user point of view, but from the product development point of view too. So really the why this standardization is important is like for these kind of 5 points. It's like reliable measurements, reproducibility, regulatory confidence from the EMA or FDA. It supports the field and the life cycle management. And it's like it will help also by getting really solid technique and method and protocols, like to have the trust from the user in a very broad way. So standardization could transform complex analytics into more reliable, easy access, and regulatory-ready quality data. In our specific case today, as I said at the beginning, we're going to be focusing a lot on like multi-detector FIFO fractionation. It was one of the main things in the project, and this technique became very, very attractive for mRNA LNP, and I will explain why now. So the FIFO fractionation, it's like a HPLC-like or liquid chromatography-like technique, which combine the separation to the characterization or detection with detectors. So the first step is a separation, which instead of taking place into a column, like in liquid chromatography, it will take place in what we call the channel. You have the drawing here. And the biggest and the most important difference belongs to the fact like it's stationary phase free. So it's a very gentle separation because there is no shear forces that you could expect could damage your product, especially with LNPs, which can be quite fragile to shear forces. It makes things much easier and reliable to analyze. So separation is in a channel. You can apply a wide range of mobile phases, which could be compatible with your product to make sure you are analyzing it in a good way. In the case of mRNA LNP, you can even use the buffer used for your particles, which can be PBS or TRIS. The step 2 is the detection and characterization. The advantage here is that you can couple multiple different detectors for various measurements at once. So you get a higher knowledge and detail on your product. If we look at the technique outside LNPs, there is a very broad range of applications, from like vaccine and gene therapeutics, as we are talking now, but like a lot on nanopharmaceuticals, protein, biopolymers, synthetic polymers. So it's like very much used, and it can be applied in many ways. Getting back to the detectors, so it can, FFF is like providing the separation of the LNPs, and then it reaches the detectors. And here you have a bit of a summary of what you can get out of that. And as you can see, you can get a lot of different measurements at once, meaning like with the light scattering techniques, you can get the particle size, the particle distribution, the particle number, looking at the integrity of the particle, looking at their morphology and shape. And then with the absorbance, UVVs, or refractive index, you will look more into their concentration and composition. So in an hour, more or less, you can get a lot of CQA's answer. But then the question became, when we were working on the project proposal and along the project, like, how do you end up with like reliable protocol? And that's where it gets very interesting to try to see like how to standardize it and to run a ILC. So now I hand over to Enrica, that will talk about the interlaboratory part of it. But before that, I think, Alex, you want to have a second poll to see what people are thinking of some part of it.
If I may, let's ask people the right question. So the question is about indeed variability across labs before Enrica shows us the way. So in your experience, what most often drives differences across labs? You can select 3 choices. Incomplete reporting of, let's say, materials and methods, if I may. In paper, also in technical notes. Raw material variability, lipids, buffer, consumables, sample prep and handling. Instrument setup and method settings, the methods clarity, the SOP clarity, or operator training. So it's about clarity and skills at the same time. Standards, calibration, control that simply doesn't work or doesn't fit with your own workflow. And finally, data processing or choices that you have to do for the reporting. Or you can simply be very honest and claim that you just don't know. So yeah, please, we'll take 1 minute. You're really fast to answer these polls, guys. Thank you for that. Well, we have a quite evenly balanced, let's say, situation, except that sample prep and handling is still a mess. And it's still very complex. But also the problems of accessibility of fully described materials and methods. We know it's a problem. But also the rest is quite evenly shared between raw materials, SOP clarity, standards. So yes, I mean, all of that is an issue, but maybe sample prep and reporting in materials and methods are even worse than the others. So now maybe Enrica, you can give us, put some light on this blurry landscape.
With pleasure. So let's go on about the interlaboratory study that has been organized in the Metrino project. I just would like the next one. Yes. Just to make you short overview, so the idea in the project was to develop fractionation and separation strategies for some, on one side, the characterization, but on the other side, also the separation, as you pointed out, the problem of the sample preparation. So to use these fractionation to separate nanomedicine from biological matrices. So in this sense, several techniques have been tested, but finally it was the most part of the work was focused especially on asymmetrical flow fractionation coupled to different detectors, especially coupled to static and dynamic light scattering and UV detection. And we had some aim to have good recovery and good uncertainty associated to this kind of measurement, especially aligned to the ISO technical specification 21362, which asks for recovery higher than 70%. So we will see now in a short time the result of this exercise. Just before, to outline that during the project we implemented some methods for different objects, actually not only lipid nanoparticle, but also metal oxides and liposomes. And so here we focus especially on LNPs, and we obtained a very good result, especially these fractionation methods separate NMP from the matrix without the need for more invasive extraction methods. And that's a very good point, because you know that when we need to apply extraction method to extract nanoparticles from the biological matrices, then we have some transformation, and it's more difficult to interpret the results. So this is very good point for this kind of analytical approach. So that's why then we thought that it was good to go through standardization. So why use interlaboratory comparison for this kind of analysis? First of all, because we are in front of fragmented analytics. That means that we use in different labs, different instrumental configuration, we define different measurements, and we treat the data in different ways. There are regulatory needs. You know very well that methods should be robust, validated, and comparable. And from the point of view of the metrology, we need to clearly define the measurements. We need to establish the uncertainty associated to the measurement, the traceability to the international system, and to evaluate the performances of these methods through interlaboratory comparison studies, using, in the best case, when they are available reference materials. So this repeats a little bit what Jérémie already said before, but it's very important to understand that the trust requires the comparability, and this is for everyone. It's for the industry that must trust what is developed in academic laboratories. This is for the regulator, of course, and this is for the researcher itself that must trust the published values. So I think that this phrase resumes very well. I found it interesting because sometimes we put a lot of energy in finding better methods and high resolution instrumentation, but sometimes the trust will come from independent laboratories getting consistent results. Just a few words about the idea of interlaboratory comparison. So here you have the general definition, but to remind that interlaboratory comparison are organized for a different reason. Sometimes they give value of characterization of reference or certified reference material. Sometimes they are organized to evaluate the participant performances. We call this proficiency testing. And sometimes we use that for the evaluation of the measurement procedure performances. This was the case here in our exercise using the ISO 5725 part 2, which allow to establish the method reproducibility, which is a combination of the repeatability of the method, so the variability which is within a single laboratory, and the interlaboratory variance, which is the variability across all the laboratories. Everything combined together is the method reproducibility. So the experimental design of this ELC was identified, inside the consortium. So most part of participants come from the Metrino consortium, with just a few external that were involved in this exercise. And so in the end, we were Metrological Institute, research organization, manufacturers, and contract research organization working on that, 10 participants, covering different instrumental configurations. So this is very important because we have 2 suppliers worldwide today, but different channel types. We will not go inside through the technique in this sense. Jérémie showed before the general principle, but there are different setups which allow to perform the separation. And so it is important to have this idea about which is the impact of different steps on the final result. We had also, so we have here one harmonized method, but also we involved two complementary techniques, MAD-ELS and SAS. And we studied this on two NNE types produced by both SINTEF and Curadig Nanobiotics. So two aqueous batches of LNP on patro formulation that were produced last year in January, distributed to the participants immediately after the production at 4 degrees, and we gave the participants 2 weeks to analyze these objects. In parallel, there was a stability study which was organized, led by two different institutes over 4 and -20 degree temperature for 3 months, starting from the arrival of the samples to the participants. So in few words, the stability assessment was performed by MAD-ELS, and the results were very good. Here you have one example on only one of the samples and 4 degree, but you can see that the samples were very stable from the beginning until 3 months. Then we put a lot of effort in trying to eliminate as many degrees of freedom as possible. So we performed this by imposing a strict analysis sequence, defined membrane and buffer, no sample treatment, and especially harmonizing the data processing that required a lot of discussion because we have different measurements which are addressed applying these techniques, and different way also to integrate different model for fitting in molds application, and so we tried to fix as much as possible all these parameters. And here I would like to point out an example, so the full white at half maximum versus the full peak integration. These are two different ways to evaluate the same raw result. The full white at half maximum, it's a core. It's evaluating the core of the peak, so which it's a precisely defined way to work. It's in some way a robust measurement, and it's very good for comparing the performances of the different laboratory. But on the other side, the full peak, it gives you a lot of information. So this is very interesting for people which are analyzing, for the producer, for the analytical chemists which want to take the most part of the information. And this gives also the possibility to compare with batch methods as this type of integration. So this is an example, but it's important because in the way that you are integrating will give you different results, slightly different or very different if the polydispersity of the sample is important, for example. So the standardization must help to define these measurements. So now we can have a look about the results. As I said before, we have this full white maximum, and so here you have the results about this first kind of integration. We can first have a look about all the results together. On the left side, you have the results about the Curadig LNP, and on the right side, the SINTEF one. And then the color will indicate you two different setup, pre-cleaned and standard channel, which are slightly different. And of course, you have also the indication about the supplier. So if we consider everything together, we have an average here. And if we consider by configuration, we can observe that we have a significant, even if not very big, difference between the average size that we measure. And also what we notice is that the recovery is always very, very good. It's higher than 90%. So to have a general overview, then we have applied the ISO 5725 which allow also by applying statistical tests to eliminate few laboratory when they are too far from the average value or they acquired too dispersed results. And in the end, so we can give an average value without the excluded labs and the performances of the methods that you can find here. You have the reproducibility, the repeatability, and the interlaboratory variation. So as we say, the recovery is very, very good. And for the size measurements, the repeatability was excellent, 1 to 2%, and the reproducibility is also very good, lower than 7%. So what we learn from this exercise, that this approach is robust for lipid nanoparticle characterization. On one side, we know that we can perform online separation from the matrix, and we obtained high recovery, excellent repeatability, good reproducibility, and we pointed out also that the measurand definition is very critical. By the way, these results will contribute to ongoing standardization activities under the ISTM and also under the ISO TC 229. And we are also going to publish this result. So I will now give back again the floor to Jérémie, but before discussing about, because just I would like to say one point. So we discussed here about, what is happening? We discussed here about the size measurement. And so now the question is, we show that we still have some variability in size measurement under strict harmonization. And so now have a look about what happened when we move to RNA concentration, purity, and integrity. And before, some exercise for you. Poll number 3.
Yeah, let's talk about the mRNA payload a little bit now. After that, we've discussed about this ILC. I'm launching now the new poll. So in your opinion, guys, what is the hardest to measure in your daily workflow? Not in general, but in your lab or your SME or whatever. The concentration of the mRNA, the integrity, the purity, the encapsulation efficiency or loading, the free versus encapsulated RNA indeed, some adducts between RNA and lipid, mRNA and lipid, or honestly you're not even sure about because it can depend on various conditions, it's never the same, or you just don't know because you don't measure it. Every time you're faster to answer. By the way, while you take some time to answer our poll, I just want to say, right now, the Q&A is totally empty, which is okay because you are mesmerized by our speakers. I agree with you. But in case you already have a question, don't lose time. You look on the bottom toolbox from Zoom and you click maybe on plus with the 3 dots and you see the Q&A tool. And then you can ask your question, but also vote for your favorite questions because we'll start by them. Okay, I guess now I will close and show the results. First, people are not sure. It's not that obvious. Maybe because it depends, or maybe because not all of you measure the payload correctly. But yeah, there's one clear message on the RNA integrity. And I guess, Jérémie, integrity is absolutely important because at some point we want to trigger some biologic effect, right?
That would be nice.
That would be nice.
So yeah, we have gone through a very thorough standardization and inter-laboratory comparison effort with Enrica and FFF with a lot of results. Now this part going into more the RNA, there is kind of no result. It's more like looking at the options that can be available and pointing out 2 different aspects and what does that mean? And when we look at something that can be easy and it gets more complicated, or how can we solve some questions and issue around? I will explain what I'm going through. But yeah, let's start from like API and payload. How is the RNA doing? And I think the very key message here, for if people are not so into like RNA related field, is like one break, no effect. So you want to make sure like your RNA integrity is essential, but also your RNA concentration is obviously essential because you want to assess the right dosage and the right dose into someone. So that's important to keep in mind. Then if we continue, you have a lot of other things that are important to look at it from different aspects, like the concentration, the size and the length, the integrity we talked about, the purity. Let's say that, double-stranded RNA is like immunogenic, so you don't want to have it. Like the capping and poly-A tail, if they are not well capped or the poly-A tail is not there, your mRNA is not going to trigger anything. So all of these critical quality attributes are important regarding safety, efficacy, potency, and stability. But the question is then, okay, how do you solve that? How do you look at it? And again, I want to show that, okay, you have a broad range of different techniques that can be applied from easy or seen as easy to quite complicated. And how do you deal with that? And I think what I want to show is that sometimes what I have seen in the lab is like some things that I've found that, okay, it should work because it's an easy protocol, it's an easy thing, will be easy, but it is not obviously always easy. And that's where I think I would like to see in the Q&A if anyone has a feedback, a question, or just like a user story to tell. I think for when we look at the concentration, currently the assay that is used the most is like fluorescence method, such as ribogreen. It's used everywhere. It's used in my lab, in many labs, in industry, even for released to FDA and EMA. Mostly at the beginning because it was the one available and the one kind of working. It is high sensitivity and broadly applicable. It's a very simple workflow when we look at it from the protocol delivered by the instrument manufacturer or the kit manufacturer. But then you have some issues that goes with, like, first of all, it can easily overestimate the free mRNA due to the surface accessible RNA that you will have, and then you will have like a lower, for example, encapsulation efficiency. But I think where I want to really talk about, like, it depends on a very effective LNP disruption. So you are opening up to get your total RNA free, but you also want to make sure none of the lipids are still attached to the RNA, so the dye can bind to it. And that, I think, where it gets quite interesting and challenging. Because then you might end up that for one formulation, things are quite easy because your detergent is working really well, but then another formulation in which you play with different lipids can get quite complicated to work with. But then if you have no standard or no detail around, it's quite challenging to work and provide the right results to your lab. It requires careful standardization for reliable results. It's what I meant. Like, it's not because it worked on product A that it's going to work very easily on product E. It's generally suitable across LNP types, that's true, but still formulation specific matters. So it looks easy, but it is not always easy, and effective disruption is, I think, a big question, and sometimes it gets quite wrong. But still, you have zero information about the RNA integrity because this dye will bind as soon as you have 40 bases available. So maybe everything is broken into pieces, but you still have a concentration. Then you get into like, okay, how can we deal with that? And I took the example of capillary gel electrophoresis, which could look at the same time in the concentration, in some ways, the purity and integrity of the RNA. So compared to fluorescence-related kits and technique, it's much more complicated because it's like a high-resolution separation and detection. It's a much more complicated instrument, much more expensive. But it has some very good aspect. It's like a high resolution, so you can really analyze from like 50 bases to like 10,000 bases or 12,000 bases RNA. It's quantitative and provides some structural information. It's very suitable for QC environment because it is already used in other fields. And you get a much better insight into integrity. So that's really the key point. You can quantify your intact RNA instead of all. But you dig into the same issue. You need to have very good RNA extraction and LNP disruption that will not affect the traveling through the capillary in the CG to reach the detector. It can be quite challenging for encapsulation efficiency because traveling through the capillary can disrupt some of the LNP, so the free RNA can be a bit wrong. It is sensitive to matrix effect, and obviously it's more complex and time-consuming. So again, it is complicated and a long protocol. You have the question of the effective disruption, and it's sensitive to matrix effect. So what I want to say is that the RNA concentration, which can look like something easy at the beginning, concentration and purity, it's still a challenge in the field. And the composition dependent of the LNPs in terms of lipid, but also in terms of RNA, when you start to play with different chemistries, the dye might not bind the same way. The RNA might be more complicated to work with. So it goes with many other challenges. And in that sense, I think that field will require also quite some interlaboratory comparison to see what can we do to get reliable and very solid protocols to look into these questions. And I will be very curious to hear point of views from people about these parts. To summarize, because I think we are a bit even over time, quick conclusion about this webinar. I think point one, we saw that multi-detector AF4 proved to be a very robust and reproducible method when, like, across such a large range of laboratory, it shows excellent result for RNA LNPs. Inter-laboratory comparisons are essentially demonstrate method comparability, evaluate performances, and build confidence, and that's really the key, and that's the success to continue working on this method and working on RNA LNPs with FFF, for example. Third point, standardization require clear measurement definitions, very solid harmonized protocol, and very often it's even much better when you have shared reference material to work on. While the particle size measurement can already be harmonized, as we have seen now with FFF, we see that other aspects can be still a bit challenging, such as what I showed on RNA-related attributes. And finally, I think the Metrino project has been really nice and is still really nice. It shows that collaborative work throughout such project can be the key to establish reliable measurement framework protocols on dedicated products. Today, we only talked about RNA LNPs, but we have also worked on liposomes or other products. And such projects are really the key to work together and to gather solid results. And finally, I think it's really key to say a big thank you to all the partners in this project, which many of these results come from all of us. So yeah, thank you, everyone.
Thank you, Jérémie. Thank you, Enrica. Every time I say it, but I've learned a lot again and again. Really, fantastic way to show important results. So now we'll start the Q&A, but let's use some kind of an icebreaker, if I may. With the new polls, you were starving for new questions, guys, right? I know you love it. While I'm launching the new polls, please start to ask your question in the Q&A. So once again, if you don't find it, you click on plus, the little circle with the 3 dots, and you find the Q&A, maybe in your own language, but it should be easy to find. Right now, there's no questions. So to break the ice, let's start the poll number 4. So when labs or experiments disagree, what do you check first, guys? Do you check first the problem of sample preparation and handling, instrument method and settings, data processing, or standards calibration controls, or maybe the material is really by itself heterogeneous, or honestly, and you can be honest, we don't judge, I rarely compare across labs. So your first check, you have only one possibility to answer, Jérémie. We've been extremely tough on this one, Jérémie. Just one possible answer. Let's be drastic. Extremely clear. So everyone say, first I check the sample prep and handling or the method and settings, but not the standards, not the quality of the material, and not the data processing. Do you have any comments, Enrica or Jérémie, on this first impression, picture?
I think it's not surprising. I tend to agree that the first thing can easily be the sample prep or handling, because you can mess up something. And so I'm not surprised. An instrument method or settings, instruments can be the same, but not exactly the same, and so on. So in one way, I'm not so surprised about the answer. I would have been curious to remove the sample prep to see what would have come next, because I think it's kind of interesting to see, like, our standards and calibration and controls used. People might not be comparing things. Not surprising, to be honest, but interesting.
Enrica, any additional comments on this one? Now you can unmute.
Thank you. No, I don't want. Especially, I wanted to have back my sound in case of. But in this case, I cannot say more than what people said in the poll and what Jérémie completed. So it's not surprising.
Do you think, both of you, that at least there's something that people never check first and should be checked, not just sample prep or the instrument by itself?
But I think it's reasonable that they started to check these, because it's what we do also. I mean, in the lab, first we are thinking about. I mean, if we have reference material, if we have standard, then we are more sure about what we did. But in case that we have not, of course, then we are starting to thinking about all the parameter, where it can come from, the problem and everything. So I think that this is normal.
Oh, yeah, maybe a first question, which is probably for Enrica. Let's talk about the interlab comparison. I mean, the setup is extremely strict. I mean, it's extremely rigorous. You have really strict harmonization. You clearly say we want to erase the maximum possible freedom to diverge, if I may. And still you see some variability. So what should that teach us about real-world comparability? Isn't that a bit discouraging? Like, even in the strict condition, you see things that vary a lot, and how to handle this some kind of intrinsic variability?
To be honest, I have the impression that this variability is very good, because you know, sometimes I work also on other fields, and I really see the difference when the methods are not yet mature. And so in this case, we are not talking about 50% of reproducibility coefficient of variation. We are talking about lower than 7%. So in my opinion, this is very good. And I think that, yeah, for this kind of measurement, this kind of setup, I'm not expecting to go really to more strict values. It is normal to have some variability, because we are not really looking, we give average values, but we are looking to detailed separation inside the channel. So it is normal to have some degree of variance between the different setups and different labs. And if you consider another point important to remind, and it is typical of nanomedicine, I mean, all these objects are also influenced by the conditioning and the temperature, because in other fields, in nanometrology, we work with objects which are more stable, and here there is also this, that they are quite fragile in this sense, and we were very happy in this sense also about the stability study. So I'm quite happy about this exercise, personally, from my point of view. The point is to address all the other measurements, all the other parameters.
Yes, indeed. I'm also very happy about the results. It's just that, let's say that. So it's normal if you show some variability, at least you should compare it with other labs. But maybe my second question also is for you, Enrica. Yeah, I mean, now we have the money, the time, the expertise in Metrino to engage in such ILC. But I really love the fact that you say that it's not just about having the fanciest high-resolution instruments, but you need to have consistent results with other labs. But when you are not in a European project for metrology, what is the best method to compare concretely in a nice way with other labs? I mean, is that easy to create collaboration on that? Are people eager to do so? Should you contact a metrology institution from your country to help you build this kind of comparison? How to start to compare more often, not just two publications, but your results in your lab in a more consistent way? What is the practical way to build that as a culture?
The practical way, okay, in other fields which are more well assessed and less novelty fields, I would say, it is normal to have really a structure which propose proficiency testing to the laboratory in order that they can challenge their capability and prove their skills. So this structure, we will need this in the future. It should come. This is, for me, it's parallel to standardization development. Then we can have material. When you have reference material, then you can have also organism organizing this kind of interlaboratory test. And if you remember in the slides where we have the different scope of different interlaboratory comparison, we have also one way, which is proficiency testing. And so this is really to evaluate the capability of the lab. I think that this is the way forward.
Great. Maybe a last one, which is more on your part. Well, no, basically also for Jérémie, but I also love the fact that somehow separation can rescue interpretation is a great line, but but it's extra effort, right? So how to decide if it's worthy or not, when to use that or not?
Jérémie, you want to take this one?
Go ahead. You were there already, so go.
But because I have not understood the question, but you don't save me, so I can ask Alex to repeat.
You want me to repeat the question, maybe?
Yes.
Yeah, exactly. The question is simple. How do we, on which criteria should you opt for fractionation first? Because it's more efforts, indeed you have better results.
You mean compared to other techniques like, for example, just light scattering? So yes, these...
The investment and the return of over investment of fractionation.
Fractionation is not only a way to characterize the size of objects. So first of all, even if we remain just on the size topic, what we can keep in mind is that this allows to have much more information about the whole sample, because it is a detailed separation, a detailed characterization compared to other methods which are less expensive, of course, but that in some way can hide a lot of information related to the sample. This is the first point. But then the fact that we couple to different kind of detector allows so it is right that in the beginning you need more, maybe more energy to develop the method, to have the system, the skills to make it working. But once that this is done, then you can, with one analysis, maybe long one, it's chromatography, so it's not 2 minutes, but it's more like 40 minutes. But then with this analysis, you have a lot of information. And also beside the size distribution, as Jérémie said, so this is the power also, and you can create the data and catch out a lot of other parameters. So this is the force, actually. For me, it's not a kind of routine method that you use at the first step, but it will become in the future a good techniques for also really for better understand information about sample preparation in the beginning.
So would you, in a nutshell, consider that beyond state-of-the-art as a harmonized method for RNA-LNP or close to state-of-the-art?
State-of-the-art.
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 Okay, Jérémie saved us this time. He jumped on the answer, and that's... Now it's high time for Jérémie to answer the two first questions that we have. So first, I would like to thank Asadu Rehman for the first question. Thank you so much, because it disproved the fact that it doesn't work, which is kind enough. Thanks for your presentation. What's your take on the choice of mRNA extraction method to ensure reproducibility?
That's a tough one in some way, but the easiest answer is that the choice of is mostly dependent on the product you are working with, meaning the mRNA and the lipids. But behind that, I will say that it's always the same. It's much better if you can compare different ones and see, are you getting the same result, yes or no? And you might find that for your specific product, a specific protocol, which will work much better than another one that is used So obviously it represents like much more work and much more test and optimization, but I think it belongs really to optimizing and testing, and also to check the reproducibility with the same protocol to find the exact one for your product.
Okay, next one. Many thanks for the presentations. Do you consider to be also important for the future the providing of quantitative thermodynamic and kinetic data for the identification of ligands, which have an appropriate biological effect on a validated target biomolecule? It comes from our dear colleague Speranta Tănăsescu from Romania, who's, by the way, an expert of this field.
Yeah, it's a very now given you said, I understand that it is an expert. Yeah, a very key point. I think it represents now a bit the future of better understanding such product, because when you inject IV, what's gonna bring your product to the targeted cells and organs is like really what's gonna attach on, and understanding that will be the key point. So if you can do any type of like kinetic and thermodynamic in some way, but I would say mostly kinetic, of the identification of the ligands, which can be like the ligands you have been putting on yourself, or the ligands in a sense that's which molecule are going to attach in the blood, will really be the key to ensure that the product you are developing is gonna work when it goes into the blood. But that also goes with having proper protocols in terms of like how can you play with plasma and blood, how can you measure that, how can you assess the ligand, like how are they binding, are they available? I think it's really the key, and it will be extremely good to have validated protocols to look into that.
Since you're talking about plasma, before we move to the next one, just a silly question, but please, I'm not an expert, so don't laugh. I know that instantly when you put in serum, or I mean close to blood, let's say, you have the matrix, and you have, let's say, any protein-rich media might change the result, even for DLS. What is your first move that you would recommend to any student or any researcher who's not an expert, who see that the size instantly changes a lot? How to deal with that?
I think it depends what you are mixing with and in which sense it's changed a lot. I would say if it goes from 100 nanometer to 10 nanometer or below, you could wonder like is the interaction they have with the proteins can be disrupting the LNPs. I really don't believe in that, but that's an example. The other way around can be concerning. Like you can create aggregates for whatever reason. The proteins you are playing with will kind of create big aggregates. Very often what you can see is you can maybe be able to assess that you have corona formation around your LNPs. And that's what gets quite interesting in terms of understanding that stuff are binding on. And the next step is assessing which protein are binding, because that can trigger where they could end up when they are in the bloodstream.
Yeah, so sometimes that's something to understand that is interesting in variability, right? Okay, not just a constraint. Brinduza Dragoy, always asking nice question. Thanks, and hello, Brinduza. Hello, interesting meeting. I do not work on RNA, but I would be interested in the reproducibility of the biological effects of the encapsulated RNA. It happened to hear about some problems related to this aspect. I'm wondering why this happens in the physicochemical properties why this happens if the physicochemical properties are reproducible. Do you have any idea on that?
We just finished running a kind of a interlaboratory comparison on potency assay. So it's exactly what I think she's asking on. Like we shared the same kits, the same cells across, don't remember, 8 or 9 labs. We gave to these labs the same LNPs. And the goal was to check, okay, can we get the same results? The truth behind is that for some it worked, for some it didn't work. And the question became why? And it's kind of what you shared in your poll. Like, is it the sample ending? Is it that or that? And we don't have the full answer, but it is extremely important what this person is asking, because in vitro assays, again, can be seen as very easy and giving result, whatever, but how reproducible and they can be across labs, that I think gets into other issues to the link to the biology aspect of cells or things like that.
And may I ask, we are talking about which physical chemical properties, because that's also the point. Are we measuring exactly everything that we need for, or there are some parameters that are maybe less? I'm thinking about surface properties, about I don't know, because for example we measure data potential, but is data potential really representative of what is happening at the surface of one of these objects, especially when it is in a biological metasis? Maybe we have not all the good parameter for that.
Yeah, but still, I think it's interesting to think that when you have the size, the RNA loading, and so on, which could be the same, and then you end up with very different biological results, it shows, as you tend to say, that it might be behind these classical things we are looking at. Maybe there is something we don't look at, which might be very different. And one of the aspects that the field is kind of pushing to is like, how is the internal structure of these LNPs? Can they be changing over time? Can they be changing over stress when you are shipping them around, or when you are freezing them or thawing them? So there is quite some aspect that we are struggling to look into, which might play a big role into the final biological results.
Maybe two last questions. Oh, okay, so Brinduza was saying, for instance, size, loading, exactly. Maybe if I may, because we don't have any more questions in the Q&A, I would like to ask maybe a last one. And it's about the impact that this kind of project have. So we've been fighting with complexity for 3 years, trying to bring not just results, but sometimes knowledge and maybe sometimes standards at the end. So my question is very simple, but maybe it's tough to ask to answer, Enrica and Jérémie. If you had one thing after Metrino that you would like to see change in labs, some bad habits, for instance, something that imagine PhD students starting in a lab which is a lab of nucleic acid, not a nanomedicine lab. They want to encapsulate nucleic acids, and they say, I don't know, I want to measure size, and something that you often see that is really a wrong thing to do, that is not reliable, something that let's be frank, something that you hate to see, but you very often see. Is there something that you would like to see change, very simple, on the bench on the daily basis?
Yeah, but it's not so politically correct. But I think we should stop believing too much into
in the replay.
Yeah, I think my take on is mostly not always believing into the batch DLS straight, doing nothing else. I think batch DLS is a very interesting thing. It gives you information, but it gives you, I think, just the tip of the iceberg.
And what would be the next move if you don't rely only on that? The next method you would like to be more used or more systematically used?
Yeah, I'm a really bad guy, like I'm stamped with FFF. Field for fractionation.
I think we are both on this side, so it will be an orientated answer maybe, but we believe in the strength of this method, actually.
Now, we are not the only one. When you see how much industry is using it now, it means that everyone kind of understand. But I think it's something that I understand that DLS is not, to me, the numbers they give you can be true, but can easily be wrong, and not just taking them, rolling with that.
Fantastic. Enrica, do you want to add something on this first, the thing that you would like to see change on a daily basis, on a routine basis?
I was actually thinking to the same, but I didn't want to say this because, I know that we are partisan of this technique, but it's not really, it's for a reason actually, because I hope that in the future it will be more easily accessed also for the people working on the field. I know that not everyone can have this kind of equipment, but we can offer a service for that, So I hope that this interaction will be more easily in the future.
The kind of output that I was expecting, we are trying to create not only standards, but a culture of metrology. Yeah. Things that you should think of, even if you're not an expert, you should contact the people that know how to do it. Last question for you, Enrica. Okay, you've talked about this ILC. How will it be made available to people? I know that you push it to standardization bodies, maybe, but how can people read it? Is it just a publication? Where, when? Do you know already what will be the final embodiment of the ILC?
Yes, it's a publication, that's for sure. I think the publication is the right place to have the time of word, I would say, to go inside in all these details, because, for example, this presentation of today, we just presented also the top of the iceberg. We have all the sex measurement, we have all the medialist measurement, we have the measurement of FFF coupled by two DLS also, which were performant, and we had not the time to present everything. We don't want also to submerge people with a lot of data, but these will be very well presented in the publication. I think that's the best, more democratic approach. So maybe open access.
Maybe. So not just the integrate the signal, as you show. Okay. So let's wait for the final output of that. Guys, I really want to thank you. I would like to also thank a lot Clémence Nadal that organized the meeting perfectly. Thank you so much, Clémence. We'll close right now. If you want to contact for extra questions, I'll be happy to send your question to the speakers if there were too non-political. Even politically incorrect.
Even political.
It's free. We are just not just a bunch of experts, but this is a place for dialogue, Metrino. So send us our question if you need, and the replay plus some insights, very practical insights, will be made available next week on the website. If you want to learn more, be in contact, just go on LinkedIn and click on follow Metrino. For us, it's super useful to share what we do. And with this, I want to thank Enrica and Jérémie again very much. That was crystal clear, very interesting. And all of you guys that made the effort to stay until the end, that was really, as always, very stimulating. And with this, I wish you a fantastic end of the day and a nice end of the week. See you next time. Oh, by the way, what is the date, Clémence, of the next one?
26th of March.
26th of March. Let's talk about correlative imaging to see nanoparticles in biological tissue with fantastic correlative imaging methods by Leonardo. See you guys. Have a nice end of the day. Bye-bye.
Thank you. Goodbye.